Hi, welcome to a Physionic detailed study analysis. Today we're going to be discussing LDL particles or low density lipoprotein particles and their effect on heart disease risk. Is there an increased risk of heart disease with LDL, with increased LDL? But additionally, on top of that, we're also going to be looking at the sizes of LDL. So looking at small dense LDL particles versus large buoyant LDL particles and see if those have an effect and if one is maybe potentially worse than the other. Because you'll sometimes hear people say that, well, LDL may be raised, but it's of the large buoyant type, and that isn't a concern. So we'll look at some data on that as well. So we're going to be going over 10 different studies to come to the conclusions that we will at the end of this video. I'm also going to touch on oxidized LDL, but I'm not going to go into too much depth on it, mainly because I'll probably do a separate investigation on that, as well as looking at triglycerides and a few other metrics as well. So if you're in for the long haul, then welcome to this Physionic detailed study analysis. We're also going to go over the mechanisms and what LDL does and why we might have different sizes of LDL and things of that nature. So there's a lot of information in this video. It's really comprehensive, but certainly if you don't want to listen through the entire thing, then you can jump to the end of this video where you'll get a really concise summary, but it's certainly not going to cover everything that I've gone over probably for the next hour or more. So just be aware of that. So if you're in for all the details, then stick around for the entire thing. Okay, with that preamble out of the way, that introduction out of the way, who am I? My name is Nicholas Verhoeven. If you're not familiar with who I am, I'm a PhD candidate in molecular medicine, and I hold my master's in exercise physiology. I've been a scientist for a number of years now. And this is what I do. I read research, I break it down into more understandable terms for you, and I actually show you the data so that you can come to your own conclusions on how you would interpret the data. So you don't have to take my word for it. You don't even have to take my conclusions. You can look at the data for yourself, and I supply you with the notes as well as a bunch of other information for your benefit. Okay, with that, let's go ahead and get into this. All right, so these are the topics that we're going to be going over. I told you that we'd be going over a background on LDL, the sizes of LDL, and the function of LDL, as well as why is LDL supposedly linked to heart disease, what's kind of the idea behind that, as well as then a comparison between small, dense LDL and large LDL. Do they have a particular heart disease risk? Is one worse than the other? We're going to try to come to some conclusions on that. And then if you're part of my insiders, we're also going to go over some strategies for lowering LDL and as well as probably SD-LDL, so the small dense LDL, because that's the one that's usually, you hear a lot of people say that small dense LDL, at least at the point of this recording, that small dense LDL is the one that's more dangerous and large LDL is not dangerous. So we're going to go over strategies to lower small dense LDL. Then also one of the reviews discussed a threshold of risk that they've assessed. So if you have a certain amount of SD-LDL, it's okay. And anything above that threshold, it's worse for your health. So I'm going to go over that as well. And then there's also another review that goes over a few of the genes. So you could do an actual, you could get a genetic test done, which those are widely available nowadays. And you can look for these particular genes that I'll have listed, and they are linked to having higher SD-LDL, so small dense LDL. And then also a mysterious triglyceride effect. I'll be explaining that and what the data shows on the effect that triglycerides have on LDL and SD-LDL and overall heart disease risk. So if you're interested in accessing all of these as well, then certainly jump on over to my Insiders program where you'll get access to all that. And if you're just interested in these three topics, let's move forward in the publicly available version. Okay, so these are the studies that we're going to be going over. Let me remove myself for a second so you can see the titles. These are all the studies. These are all the numbers of the studies because I'm going to have my notes for you so that you can look up these studies, look at the conflicts of interest, look up the funding sources and all that. I've got that all listed for you so you can download the document. But these are all the numbers of the study so that you don't have to remember the exact title of the entire study. Okay, so that out of the way, let's go into understanding LDL. For that, we're going to go over these studies. So that's study 171. And you can see I've got a little bit of a description on the funding as well as the association industry. Some of these, like I've gone extremely liberal with the idea of industry ties, but I still want to present that there are industry ties. For example, 171, just one of the researchers received lecture money from a pharmaceutical industry. So he gave one lecture and he was paid for that lecture. And that's all the ties that he has to industry. So not exactly on the payroll or anything like that to any great degree, but still there are some industry ties. The other researcher had no conflicts of interest, so no ties to industry or nutraceuticals or anything like that. So on the other hand, you have the extreme over here, study 178. They're all industry researchers. Their salaries are actually paid by the company that employs them. So if that's a concern for you, if that's something that you just want to dismiss that study, that's entirely up to you. But I'm just disclosing. Some of these are publicly funded, meaning that it's just NIH grants, usually NIH grants or some sort of Veterans Affairs grant or something along those lines. So no strings attached. You just get the money after you put in a public grant, and then you can just do your research. And there's no additional aspects that go along with that. Internal funding, this one was at a school of medicine. So, I mean, there's just a bunch of different associations. This one is the final point that I wanted to point out. And for more details on all this, again, that's all listed in the notes that you can download. So you'll be able to see which companies that they were affiliated with and in what capacity there were conflicts of interest. For this one, multiple associations. So the study itself wasn't public, it wasn't funded by industry necessarily, but there were multiple researchers that either did consultancies or gave lectures or whatnot with different industries. Not exactly extremely worrisome to me, but if that again sways you, totally fine. You can dismiss the information that's related to this study. And I will be breaking them up so you can tell which information comes from which study. Okay, so this first section of information comes from a grouping of all of these studies that I've put together, all their information, and we'll discuss how LDL functions, understanding LDL as a whole. And some of this also just comes from what I've learned through my medical school training, PhD medical school, just to be clear. Okay, so let's discuss a little bit about LDL. So LDL is this molecule right here, this, I suppose I should say particle, this low density lipoprotein. And this LDL contains triglycerides, it contains fats, and it contains cholesterol, which is different from, they're often used interchangeably, using the word molecule of cholesterol as LDL, and they are very closely related to one another. That's why they're used interchangeably. But really, this is a capsule that is made up of cholesterol and is made up of protein and is made up of phospholipids. But the core inside of here it contains triglycerides and cholesterol. And we have these nutrients that we consume. So let's say you consume cholesterol, let's say you consume fat. Cholesterol, there's another mechanism that regulates the amount of cholesterol that's actually released back into the bloodstream. But let's say you were to consume triglycerides or whatever, eventually ends up in the liver, where the liver is just like this processing system. It's like a processing plant. To me, it's the coolest organ of all the organs. I've said this multiple times across my physionic journey, just because it has It has so many different functions, and it can self-regenerate, which I think is just so freaking cool. Anyway, so nutrients like fats, cholesterols, etc., all the foods that we consume end up at the liver, and then they're processed in one way or another. And through this processing, through these hepatocytes that make up the cells of the liver, you get the eventual production of what are known as VLDL molecules, which are very low density lipoproteins. And they're extremely similar to LDL. They're just bigger. They're bigger, and they contain triglycerides and cholesterol molecules. I'm going to make that distinction from now on, that when we're talking about LDL or VLDL or IDL, which is intermediate density lipoproteins, I'm talking about the particle. And when I'm talking about cholesterol, I'm talking about the molecule cholesterol, which is found inside, like a cargo ship. It's found inside the cargo of these VLDL. And what happens, it gets ejected from the liver. So the liver packages up cholesterol molecules plus triglycerides, these fat molecules, and packages them into VLDL and then kicks them out into circulation. Then VLDL circulates through the body as well as IDL and LDL, and ultimately interact with the tissues of our body. And when they do that, they start to release and deposit, essentially deliver cholesterol molecules and triglycerides to these different tissues of the body. And as such, then they start to shrink in size. And as they shrink in size and they have these different modifications that occur to them, they go from VLDL to IDL to LDL. And the liver, however, can also eject not just VLDL, but can eject IDL and LDL. So I don't want you to think that it's a linear process. VLDL, therefore then IDL, then LDL. They come in combination, we'll say that. But as they interact with the tissues of our body, they get transformed to these different versions. Now, LDL can also be sensed. Not only does it go to the tissues of the body, but it can also be sensed by the liver. So the liver has these receptors, and we'll touch on that a little bit later on as well, that the liver has these receptors called LDL receptors, real creative, but it's at least it's functional. It's a functional name. And they bind to the LDL and essentially take them in. I'm going to show you an image of what that looks like, but essentially take in LDL, and then the liver can regulate and essentially sense how much of these lipoproteins is found in circulation, because you don't want too many of these lipoproteins. Well, we'll see, actually, but that's the commonly understood idea, that you want a certain level of these lipoproteins, and too much can be destructive. So the liver is the one that controls that, and that's one of the ways that it controls the release of these particles, is by controlling the amount of cholesterol molecule that it actually packs into these particles. So sometimes it'll just eject cholesterol into bile and just get rid of it. But that's in a functional physiological system. Anyway, so LDL can be sensed by the liver, and it goes around and interacts with these tissues of the body and delivers cholesterol molecules and triglycerides. Now, additionally, eventually the tissues of the body can also ultimately interact with HDL and can do what's known as the reverse cholesterol transport. So this is the cholesterol transport, cholesterol molecule transport, and this is the reverse cholesterol transport, where these tissues will then essentially, it's like a return policy, like a business return policy. They return some of the cholesterol in HDL packages, which are much smaller than even these three, and will ultimately get delivered back to the liver to be reprocessed, to be either ejected or to be brought back into VLDL, IDL, and LDL. And it just continues. That's just the process. It's just a circular system going back and forth, and it functions very well. Okay, so that's the function of LDL, and that's why we have these different, one of the reasons why we have these different sizes of lipoproteins. But we're going to go into the sizes of the LDL specifically in just a little while. Okay, so now if we then look at the vessel, the actual you have this tubular network throughout your entire body, which is known as the circulatory system. So you have arteries, you have veins, venules, arterioles, capillaries. You have these different systems that are essentially just tubes. There's a tubular network that allows you to maintain, for one, blood pressure. So if you have an opening in that tubular network, like a cut or internal bleeding, then the blood goes into other areas of the body that it's not supposed to, and you start to lose blood pressure in this tubular network. So that's why blood pressure is incredibly important. Now, this isn't about blood pressure necessarily, although I suppose indirectly it sort of is, but we're not going to be discussing blood pressure to any great degree here. So we've got a section of this tube. Let's say we've taken a tube out of our arm and we've put it to the side so that we can actually look at it. And then we cut it down the middle so that we can actually see the inside. So this is a cross section of the tube. Then we see that there's this open area here called the lumen, the opening, and then it bifurcates. So we've got this fork that occurs. So the tube continues down this way and it continues down that way. We're just not picturing the rest of it. Now, if we were to look at the tube and look down the center of the tube, so turn it from its side to actually looking down into the tube, we would see this. So it looks just like you would on when you look at a hose. Hose is just a tube, and you look down the hose and hopefully nobody turns it on because it's going to go straight in your face. So this tube is surrounded by a number of different types of cells. So the inside is completely clear. That's where the blood flow goes through. That's where proteins are going through. That's where the lipoproteins are going through. That's where the cells are going through, like monocytes, erythrocytes, red blood cells, et cetera, et cetera. All of that is flowing through this section, okay? So it's all flowing through this section. And the lining of this tube is first defined by these cells known as the endothelial cells. Now, these endothelial cells are a single layer of cells that essentially control the size of the tube. So they can convince other cells down here, which I don't really have pictured because I didn't want to overcomplicate this, but you have other cells that are down further down below the endothelial cells, and they're called smooth muscle cells. So these smooth muscle cells are influenced or controlled by the endothelial cells. The endothelial cells will tell the smooth muscle cells by molecules like nitric oxide, will tell the smooth muscle cells to relax or to constrict. When they constrict, the tube here shrinks so that this opening shrinks because the smooth muscle cells squeeze together. It's just like if you were to squeeze on the end of a hose, then the water starts to have less space that it can go through and the pressure increases. Now, on the opposite end, it can also convince the smooth muscle cells to relax, like, again, through the release of nitric oxide. Nitric oxide can then bind to the smooth muscle cells and relax this tube, this vessel. So that's how it would normally function. And you want this to be flexible. You want this to be to what's known as hemodynamics. You want to have a dynamic blood system where all these tubes, especially arteries, which are filled with these smooth muscle cells to a great degree, are able to stretch out or to constrict as necessary. Now, here's the deal though, because throughout this area, throughout this flowing of all these nutrients, all these cells, all these different things are the lipoproteins. Now, the lipoproteins, what turns out to be the case is that the mechanism that's proposed is that these lipoproteins are attracted to a layer called the intima. So you have this layer underneath the endothelial cells called the intima. And this intima is made up of, if you'll allow me, proteoglycans. Proteoglycans are simply, well, they do a number of different things, but for the basis of this discussion, we'll say that they are specifically structural proteins. So they offer structure for this entire vessel. So you have the endothelial cells, the monolayer of endothelial cells. Then you have the intima with the proteoglycans, and then you have this much thicker layer of smooth muscle cells which allow for the contraction and relaxation of this vessel. Now, unfortunately, lipoproteins are attracted to proteoglycans, which is these proteins right here. And these proteins are, well, they're proteins plus saccharides. So they're carbohydrates and proteins that have been linked together through a glycosylation reaction. And these proteins found under this endothelial layer, even though they serve a function, they also are negatively charged. So if we go back to like chemistry and if we go back to chemistry, or if you think about like a magnet, you have a negative end and a positive end, and what happens when they get close to one another, they interact and they closely lock with one another. I mean, you can pull them apart depending on the strength of the magnet, but in this situation, we don't have those kinds of forces. So we have a negative charge that comes from the proteoglycan structure, and we have a positive charge that's associated to the lipoprotein. Now, the lipoprotein, this specific lipoprotein, the LDL, VLDL, and IDL, all contain one common feature, which is called ApoB. This ApoB protein has a positive charge. And because of this positive charge, you have this interaction of the positive charge of ApoB with the negative charges of the proteoglycans. That ultimately leads to all of these lipoproteins being stuck to the proteoglycans. And as such, then you start to develop what are known as eventually fatty streaks. And then you have the progression from there. From fatty streaks, you have eventual immune cell infiltration, or more immune cell infiltration. You'll have the oxidation of these lipoproteins. I'm going to go into some of that a little bit in just a little while. But ultimately, the idea is that it pushes the endothelial layer up, and it continues to build over like more and more and more until the point is that this opening shrinks. So this is when it's completely healthy. This is wide open, completely nice and circular. It's kind of not like they're all circular. They might be a little bit oval, but eventually the point is that you get this encroachment on this open area. And that's really problematic because then you have the same amount of blood and the constituent parts that are in that blood that have to flow through a smaller area. And again, if this continues over decades of time and it continues to build and build and build, eventually you have an occlusion that could be only 10% open or 5% open. And then you really have some serious issues. And that's where stents come in. That's when all kinds of different problems can occur. Obviously, heart attacks can occur. So it's a progressive disease. I'm not going to go into the progression to any great degree here, but I do want to point out the commonly associated role that these lipoproteins have with the proteoglycans and the eventual outward bowing of the endothelial layer, and eventually the immune cells come in. You have the oxidation of lipoproteins, and then eventually you have the deposition of calcification. You have a fibrous layer that gets overlaid on top of this. And then you have, I just said I wasn't going to go over it. There's a lot more details I'm leaving out, but this is just kind of the big highlight points. And then once that fibrous layer gets deposited, then you have the breaking of that fibrous layer, and then you have a thrombus that gets released. That thrombus is like a clumping of many different molecules and proteins that flows through your circulatory system and can find itself downstream somewhere in a much smaller vessel that isn't large enough to accommodate that thrombus. It gets stuck there, and then you have extreme pain, and you have potentially death, or you have a stroke. If it gets stuck in your brain, then you have a stroke. You have an ischemic stroke. Or you could have it stuck in your heart or around your heart, which could lead to a cardiac arrest. Okay, so that is the association between these lipoproteins and cardiovascular disease, or the overall growth of cardiovascular disease. But keep in mind, it's not like these proteins are just around to be negative. They have positive effects. They have a role in our body. But let's examine the relationship of LDL and heart disease. All right, LDL and heart disease. For that, we're going to begin by leaning on this extremely comprehensive review. This is extremely long, but also incredibly interesting. And for this, we're going to be so right now we're just creating the relationship between LDL and heart disease. We're not looking at the sizes of LDL yet. We're going to return back to that. But for the time being, I just want to create this link first, and then, or to investigate the research that's been done, creating this link between LDL and heart disease, and then we'll go into the actual sizes. All right, so we're going to focus on this up here. So they looked at, they put together essentially all these different studies, meta-analyses, along with observational studies. So observational studies and genetic studies. We're going to go into the genetic studies in just a little bit, but the observational studies are just longitudinal studies, essentially, if that's, like prospective or retrospective. Typically they're prospective, meaning that the researchers define what they want to look at, and then they just follow people 10 years later. So they'll take measurements at baseline, let's say year 0, and then they'll go 10 years later, they'll make more measurements on those people, as well as measure how many people had heart attacks, or how many people died from a heart attack, or just died in general, all-cause mortality. And then they run statistics to determine, okay, well, what's the relationship between LDL cholesterol and the risk of heart disease? So in this situation, we're talking about myocardial infarction. So we're talking about heart attacks specifically. And here we're looking at, so a 1 hazard ratio means that there is no elevated risk. And anything to the right of this is elevated risk. And it also indicates how much of an elevated risk there is. So, and as you can see, there's nothing to the left. I mean, they're essentially just indicating that there is an increased risk according to observational research, which is associative. And I understand that. Please don't comment that this is associative, and therefore we can't make causative arguments based off of it. I understand that. Sometimes I get people that think like I'm completely uneducated. Like my entire PhD and master's, nobody ever mentioned the most basic statistics of correlation isn't causation. I get that. I understand. But they're just looking for this correlation. And they found, I believe the number was a 30% increased risk of myocardial infarction across 108,554 participants across a multitude of studies put together. They found that there was this increased risk of 30% in those individuals. Now, that is a correlation, and that is per increase of LDL cholesterol, so total LDL cholesterol, not looking at the subfractions, of 39 milligrams per deciliter. So if you see a 1 millimole increase in your LDL cholesterol, a 39 milligram increase in your LDL cholesterol, you have about a 30%, I believe that's the correct number, a 30% increase risk associated. Okay, just association. Now I'd like to, however, discuss the genetic studies, which I find way, way more interesting and are far more damning in the relationship between LDL and heart disease. So to look at these genetic studies, I want to lean on this particular study, which is among my favorite studies, mainly because I think that Mendelian randomization analyses are incredibly cool if they're done, and extremely powerful if they're done correctly. People consider randomized controlled trials to be the gold standard or one of the gold standards for data collection, and it absolutely is. However, Mendelian randomization trials are even more powerful than randomized controlled trials if they're done correctly. So to explain this, the researchers of this analysis looked at the data from a number of different studies and put it all together so it's a meta-analysis of a multitude of different studies. Now, those studies looked at 9 different genes. Those 9 different genes are associated with higher LDL, okay? Higher LDL. But there are people that have genetic mutations in those genes that drastically reduce their LDL. What's amazing about this is multifold. One is it's pre-randomized. They are randomized at birth. There's no scientific intervention. There's no statistical intervention. There's no jumbling up numbers and just randomly assigning people. It is just by the grace of nature that some people just have these genetic mutations and some people do not. So we can easily put the people that do not in one camp and put the people that do have the genetic mutations in another camp. The second benefit of this is that it means that these people with the genetic mutations will forever, for the rest of their lives, have low LDL, which is really amazing. The other thing is that there's no need for a placebo group. The reason why, which you would have for a randomized control trial, the reason why is because no matter what these people do, no matter what they do, they will have low LDL. That is so cool. They could smoke, they could have an unhealthy lifestyle, they could have all these different things compared to another person who smokes, who has unhealthy lifestyle, et cetera, they will always come out with lower LDL. So they are clamped to this situation, and that really isolates this one factor of low LDL, and the most extreme, that it occurs from birth. Their entire lives they have low LDL. It's not from like 30 years old, I started taking care of myself and I lowered my LDL and that now I have low LDL. No, it's just they just have lower LDL across the board. And the third or fourth, I don't know, I've lost track of the cool aspect of this, is one of the reason why I say that this has to be well done is because it's possible that you have a confounding variable known as pleiotropy, where the genetic mutation that occurs in this gene could also have outside effects. It could have effects on not just LDL. So yes, the gene affects LDL, but what if the gene predominantly affects LDL and does not, and also affects, let's say, HDL, or maybe it affects blood glucose levels? Then you have 2 variables that you have to control for. However, these researchers controlled for that by specifically only looking at genes that only affected one thing, and that one thing is LDL. So we can be certain, and there's nothing these people can do. So people might come back, like a randomized control trial, people say, well, what was their nutrition like? What was did they exercise a lot? Like all of that gets completely overshadowed by the power of their genetics. Okay, I'm harping on this because this is so cool, so freaking cool. And secondly, it is an associative study that is so powerful that it might as well be a causative study, assuming that you control for pleiotropy as one example, which they do. Okay, so to look at that data, here's a little schematic. We've got the people with the normal genes. So these are the genes. So SORT1, PCSK9, which is a really popular one, LDLR, which is the LDL receptor. And then we have HMGCR, ABCG8, and ApoE. And these are the SNPs, the single nucleotide polymorphisms. They're the mutations that occur in and around these genes that ultimately affect the gene to the point where you see a reduction in LDL. So, and we're comparing the results. So in this situation, we're looking at, do these genes affect LDL? So this is what the data is showing right now. We're testing, do these genes actually reduce LDL like they're claimed to reduce LDL? So we're comparing the values, the data collected from the people that don't have these mutations. They do have these genes, but they don't have these mutations, versus the people that do have these genes and the mutations. So they have these specific mutations. And again, the mutations lead to reduced LDL, supposedly. So, but what do we actually show? Well, when you look at people with the SORT1 gene with either one of these SNPs, these polymorphisms, these mutations, this is the zero. This line means that there's zero difference between the people that have the gene mutation and the people that do not have the gene mutation. Anything below that, anything to the left of that, means that there is an effect of these genes on LDL specifically. What we find is across the board, all the squares are the indication of the sample size as well as the confidence interval. It all gets into statistics. But the big picture is if you see a square that's off to the left, that means that there is a reduction of LDL with the mutation compared to people that do not have the mutation. And we see exactly that, especially with this one, PCSK9, RS115-91147. This SNP right here leads to a huge drop in LDL. And all of these are pretty substantial, but this one is especially substantial. Now, there's high heterogeneity here because with each one of these genes, you're going to see, of course, different effects on LDL. So, but we're going to control for that in just a little bit. So the point is, these mutations do lead to significantly reduced LDL levels compared to people without the mutations. Okay, next. Do these mutations have an effect on heart disease? Do we see reduced heart disease risk? So here we're looking at the one, it means that there's no difference. And anything to the left, there is a reduction in heart disease risk. So again, with all of these genes and their mutations, we see that there's a significant drop. Let's say for some of these, it's certainly less than others. So like the PCSK9, this top one, RS11206510, there is a reduction. It's a really strong confidence that we have in this because there's so many individuals that are involved in this study. And the effect is relatively mild, but there is a reduction in heart disease risk based off of this. Now, on the other hand, the other PCSK9 mutation, which is the other one that we looked at, it's off the chart. It's huge decrease, almost 30% drop in the risk of heart disease based off of that mutation. Now, again, there's high amounts of heterogeneity, 91.8% heterogeneity, meaning that, each study gets different values. Like each one, I suppose I should rephrase that. Each gene has different effects on LDL. So it's not like they all reduce it exactly 15%. Some of them reduce it 20%, some of them reduce it 5%, some of them reduce it 30%. It's all over the board. So there's high heterogeneity. So the researchers wanted to account for that. So this is the unadjusted model. So this is what we were looking at. So all these different mutations and the overall effect. And then here they're adjusting it not based off of the gene necessarily, but they're relating the gene to the overall to a standard drop in LDL. And when they standardized the drop in LDL to 0.25 millimole per liter, which is 9.7 milligrams per deciliter, we see that they all do the same thing, that they're all potent in dropping LDL, and there's no heterogeneity. So they remove the heterogeneity by looking at it from this adjusted model. Now, if you just want the raw data, you can just look at this, which this is already telling enough. But here they're just indicating that if you have, through any one of these mechanisms, you can see a drop in LDL, and you still see you can still see the same amount of drop in LDL and get the same effect in the risk to your heart disease risk. So you see a dramatic drop. It's about 20% roughly, maybe 16%, 17, et cetera, but around, let's say 17 to 20% drop in heart disease risk from these mutations. This is, I can't express to you how damning of evidence this is. Like, I know a lot of people push back against observational research and all that. That's totally fair, but there's so much observational research. And I'm going to go into some of the problems with the observational research indirectly, because I realize that some of the observational research indicates that there is, let me quick go back here. Okay, so I understand that some of the observational research indicates like there's heterogeneity there too, because there's all kinds of different factors that could be affecting the research. There, maybe we need to account for triglycerides. Maybe we need to account for HDL, all these different things. Maybe we need to account for weight. There's all kinds of factors. I totally get that. And I'm going to explain one of the main reasons why there is such heterogeneity in the long-term research, looking at these prospective correlational data that I briefly touched on earlier. But this data, the randomized, the Mendelian studies, that is, I can't, I'm hoping that you're understanding how powerful these studies are. They're not like other correlational research. They're not like randomized control trials. They're like a melding of both put together, assuming again that they're well controlled. And this one really was. Okay. So next, we're going to be going over these studies to look at LDL size and the effect that it has on heart disease risk. So, okay, it's great if we have elevated LDL, maybe not great, but if we have elevated LDL, that's one thing, fine. But what if it's of the larger variety? So the large buoyant type versus the small LDL, because a lot of people think the small LDL is the only one that's of concern. So LDL size, is there a heart disease risk? Again, just as a reminder, we just went over LDL itself. So that's the combination of LDL, all the sizes of LDL are combined into the total LDL number. But there are different sizes, and these numbers, the actual sizes themselves, vary depending on what kind of measure that you used. I think this was using gel electrophoresis. They ended up determining that there are more classes than just these four, but I've combined them to make 4 classes. But ultimately, you'll get a number that's going to determine if you are in a small, dense LDL or in the large buoyant LDL class. So the large buoyant LDL class is obviously, as you can see just by visual, that these two, LDL 1 and 2, belong in the large buoyant LDL. The small dense are considered LDL 3 and 4, and I've got the nanometer sizes. Now, again, those change depending on what kind of measuring technique that you use. But the point is that within the LDL class, the total LDL class, which is all that we've discussed up to this point, there are many different sizes of LDL, because as it keeps getting taken up, like the cholesterols and triglycerides and whatnot, it's going to shrink in size. It's going to do different things. And the liver can also release different size LDL particles. So the idea, however, was that the small dense LDL, now allow me to go back here real quick, that's these two, that the idea was mechanistically that the small dense LDL, and there's nothing behind me, so don't think that you're missing something. The idea was that the small dense LDL are more harmful than the large buoyant LDL because of three main reasons, but there's certainly others as well, but three main reasons. One, that they have an easier time entering through the endothelial cells into the intima and can then bind to the proteoglycans more easily. So that's one factor. The second factor was that small dense LDL are oxidized more easily. And because of this oxidation, you get more of an immune reaction. You get more of an inflammatory reaction that occurs in that area, which ultimately recruits more immune cells. Immune cells then cause more reactive oxygen species. They become activated, and they end up doing even more damage to that region, to the cells, the endothelial cells, the smooth muscle cells, as well as to the lipoprotein particles themselves, oxidizing those lipoprotein particles further. And the other thing is that because of the size of the small dense LDL particle, the protein, the ApoB protein, is the configuration of that protein, what's known as a conformational change. The structure of that ApoB protein is altered in such a way that the LDL receptor, this is the liver cell, so this is the membrane of the liver cell, this is the inside of the liver cell, this is the outside of the liver cell. The receptor doesn't have as strong of an affinity for the small dense LDL because of that change in ApoB, because it recognizes the ApoB, binds to it, and then endocytoses, that's what this process is, endocytoses the receptor into the cell, and then goes through a recycling process by getting rid of it or dumping it back out in the system, repackaging it, whatever. But if the receptor can't bind to the LDL as much, then it has two-pronged effect. One, the liver can't sense how much LDL is actually present in circulation. So it's just going to continue to produce the same amount of LDL as it normally would. But under normal circumstances, if it were taking up a bunch of LDL, then in that situation, it would start to slow down its production of lipoproteins to adjust for the influx of the LDL that's coming in, because there's an overabundant amount out in circulation, so the liver reduces its LDL production. Now, the second thing is, however, because it's also not binding, that means that there's a longer time that the small dense LDL stays in circulation. That means that there's a greater amount of time that it can continue to interact with the intima, that it can continue to interact with the endothelial cells, and ultimately, the greater the exposure with small dense LDL that you would not necessarily get with large buoyant LDL. That's the mechanisms that are proposed. I'm going to go into the oxidation and all that stuff in other content, but I still wanted to acknowledge that there is this other factor that people have mentioned in the past. But what we will try to answer is the effect that small dense LDL, regardless of the mechanism, if increases in small dense LDL, if that there's an increased risk. That's what we're trying to find out. Not necessarily if it has to be oxidized or if it has an easier time entering the intima. None of those mechanisms are important right now. I will investigate those in the future. Hey, I hope that you're enjoying the presentation so far. If you're interested in learning how to actually lower your small dense LDL and your total LDL, as well as understanding some of the relationships between your genes, so actually knowing some genes to look out for, which you can actually get tested and then find out if you have a higher risk of small dense LDL, or if you'd like to learn more about the triglyceride effect, or if you'd like to learn a little bit about the small dense LDL threshold, so what is the threshold at which risk seems to increase, then I would certainly welcome you to join the Physionic Insiders, which is a program where you get full access to the rest of this video, as well as all my other study analyses, seminars, office hours, which are live sessions with me where you can ask me particular questions, and I've honed in on those questions, make sure that I leave some room and some time to actually answer your personalized questions. So if you're interested in all of that, along with other perks, then I would strongly recommend that you join the Physionic Insiders. And without further ado, let's go ahead and jump back into the rest of the public version of this presentation. Okay, so we're going to lean on this study first, study 174. And for this, it's not the best study, to be completely honest with you, because what the researchers did is they took a number of different studies that looked at the overall small dense LDL particle number and the small dense LDL cholesterol. I'm going to distinguish between those in just a second. But they looked at some randomized control trials. They looked at some observational studies. They looked at some cohort studies. They just kind of mixed and matched as many studies as they could put together to create this meta-analysis. Is that ideal? No, it isn't. So there is that drawback of this study, but yet I still wanted to present the research, or the just so that you can actually see it. Now, the distinction between the particle number and the small dense LDL cholesterol. Small dense LDL cholesterol is a measure of the actual cholesterol molecule. Remember, that's really important, the distinction between the two. Small dense LDL, the cholesterol molecule that's found inside of the core of the small dense LDL particle. That's what this is measuring, just this. And then the assumption is, well, because we're measuring this, then we're measuring the particle number. On the other hand, we can also measure the actual particles and ignore the cholesterol that's found inside, but just look at the particles themselves. So here we've got the list of studies that they ended up incorporating. Here we've got the number of participants. Here we've got different adjustment levels. What are these adjustment levels? They are up here. So they use statistical adjustments to find out, well, so each one of these studies did different statistical adjustments to find out the effect that small dense LDL particles have and small dense particles with cholesterol. So specifically looking at the LDL-C, that C is really important, and we're going to touch on that in just a little bit as well. And they adjust for the lipid subfractions for other lipids, like let's say HDL. They might control for HDL or statistically control for HDL, and see if there's still an effect of the small dense lipoproteins themselves. They might control for lifestyle, meaning that they don't control for lipid subfractions, but they only control for lifestyle, like smoking habits, weight, maybe not diabetes, but like exercise, physical activity, those kinds of habits. And then the third one, the ones with the three pluses next to them, are the lipid subfractions plus lifestyle. So that's the strongest statistical method that they can use. And then they look at the overall effect. So again, the one indicates that there's no increased risk of heart disease. Anything to the right of one means that there is an increased risk. Anything to the left means that there is a reduced risk of small dense LDL particles. And what they find is that overall, there seems to be an increased risk. So we see that the hazard ratio, or the odds ratio, is 1.36. So it's 0.36 over 1, and therefore there's likely an effect of these small dense lipoproteins, meaning that they cause harm to the heart or cardiovascular system as a whole. Now, I looked at the supplemental material because I feel like somebody might ask about this, because they're grouping everything together. And I was specifically interested in this condition right here, lipid subfractions plus lifestyle. All right, I think, lipid subfractions plus lifestyle. And they ended up having a separate analysis where they just looked at those studies. Now, that's drastically reducing the number of studies that they have available. So the power of the analysis decreases quite a bit. But they still said that they found an effect even when they just looked at the studies with the triple pluses. So that would indicate, again, that the small dense LDL specifically have an effect. Now, what about for the cholesterol? So when we look at the small dense LDL cholesterol specifically, we find that the effect, there might be an effect. And I don't think they ended up actually reporting what they thought. So I'm assuming that they were a lot less convinced of the effect here. So it's 1.07. So they seem to think that this is, well, they also have more studies included in this. They find that this, looking at LDL particles, seems to be a stronger indication, or indicates more strongly that small dense LDL particles have an effect on cardiovascular disease. And there might be an effect with looking at cholesterol, but the effect is certainly much, much smaller. But we're going to return to that in different ways in just a little bit. Okay, the next study to look at would be this study, Study 176. For this one, they do have more data than what I'm showing here, but this is the best illustration of the data that they end up reporting. So to break things down, here we've got our reference marker, 1. Again, it's usually 1 or it's 0. Usually it's 1. So we've got 1. This is what everything is compared against. We've got our small dense, so we've got our total LDL cholesterol. So looking at measures of LDL cholesterol. So when you go to the doctor at present, when you get your LDL checked, they do this test. They do a test for LDL-C. So they're testing not for the lipoproteins necessarily, but for the actual cholesterol itself. On this side, we have small dense LDL-C, so the cholesterol of small dense LDL. Now remember, based on this study, there isn't as much of an effect as what we see with looking at the particles, but this is what they ended up using as a measure, and they still found an effect. So on this side, looking back at LDL-C, we see everything in this column is under 100 milligrams per deciliter, so lower ends of LDL levels. Everything in this column is above 100 milligrams per deciliter, so this is higher total LDL levels. This is looking at the quartiles, so increasing levels of SD-LDL-C, so looking at small dense LDL. So they're trying to find the relationship of, if you increase with each stepwise increase in small dense LDL, do you A, find a relationship with increased total LDL? And secondly, do you still find a relationship with lower LDL? Now, this is really, really fascinating because sure, if you have stepwise increases in SD-LDL-C, you would expect that there might be increases in total LDL because, again, SD-LDL-C feeds into the overarching LDL-C. So think of LDL-C as the director of the department and SD-LDL-C is a professor in that department. I'm getting real academic here. Okay, so And any number above 1 indicates the amount of risk increase. And what we find is that there's a statistically significant effect when we have the highest amounts. They don't give you the absolute numbers, but it could be like 10, 20, 30, 40, it could be 20, 40, 60, 80. I don't know what the numbers were, but the point is that there's an increase. That's all you really need to know to be able to create this association. So at Q4, the highest levels of small dense LDL cholesterol, we see that there's a huge harm or increased risk when there's high LDL-C, total LDL-C. And you would think, okay, maybe that's because that correlates. So that feeds into the high LDL-C. Maybe this isn't the actual SD-LDL-C, but it's actually just the total LDL-C that's having an effect, or at least that's running part of the effect. But in reality, if we try to dissociate those two or try to create some distance between them, where we look at lower LDL-C, and we still see high levels of SLDL-C, so now you're starting to create some separation there, we still see that there's an increased risk. It may not be quite as much, but there's still a substantially increased risk of heart disease, cardiovascular disease, when you separate them. So this would then implicate SD-LDL-C as having a separate effect from LDL-C. Okay. So that's this study. Now, if we lean on study 171, this is a scientific review, so there's no data in this one. They also indicate that SD-LDL SD-LDL-C may be a better marker, has been associated with having a greater predictive ability than looking at LDL-C. So specifically looking at SD-LDL-C is more powerful than looking at LDL-C, which is, again, where a lot of our blood measures are, LDL-C. We look at LDL-C. But here's the thing. Again, I mentioned that large buoyant LDL-C and small dense LDL-C, those cholesterols feed into this overall measure of LDL-C. So there's a problem with that. And this is where, allow me to stop here real quick, because this is the point at which I think it may not explain everything, but it explains a large amount of the data discrepancies between studies. Because a lot of studies look at LDL-C, and a lot of these reviews are starting to come to the conclusion, like these three, that we cannot look at LDL-C alone, and we probably shouldn't be looking at LDL-C in the first place, because it's an okay marker, but it's not great. And here's the reason why it's not great and why it creates misleading results. If we have, this is generic terms, let me remove myself so you can see the other image here. In generic terms, let's say we have 5 units of cholesterol. That's how generic we're going to get with this. We have 5 units of cholesterol molecule that we need to transport throughout the serum. The benefit of a larger LDL particle of the actual protein structure of the core, because it has a larger core, it can fit more LDL cholesterol. It can fit more cholesterol in that core. So you only need 2 lipoproteins, 2 particles to carry the same amount of cholesterol molecule versus if you have small, dense LDL particles. Because they're so much smaller, they require more particles in general to carry the same amount of cholesterol. So that's why looking at LDL-C compared to SD-LDL-C, which is this marker right here, which is what we typically look at, it can be extremely inaccurate, because the LDL-C, you could have the same level of LDL-C, but the type of particle that actually carries that LDL-C is extremely important, because you could have way more of these small dense LDL-Ps, the particles, to carry the same amount of cholesterol as opposed to if you had large buoyant LDL particles. So that raises this question of, if we have more of these, does that then mean that we have increased risk compared to the large buoyant LDL particles? And for that, we can look at this study. Okay, now to look at this, here they're looking at the intima thickness. This is, allow me to go back. This is this right here, the intima. So that if there's a thickening of the intima, you know that's not a good sign. And that's what they're looking at here. So intima thickness. Okay, to orient you. They're trying to segregate the people that have predominantly large LDL particles. So this specific study looks at LDL-P as opposed to LDL-C, which is the distinction I'm trying to make here. So they specifically look at the particle number, not at the cholesterol number, which is a direct measure of the lipoproteins, which is probably the real problem, not the cholesterol itself, although there are some mechanisms by which the cholesterol can be problematic, but we won't touch on that right now. The bigger problem is the actual particle itself. Okay, so what do we find here? They're separating the people with large buoyant LDL and the people that have small dense LDL, and then the people that have a combination of the two. Here are the N, so this is the number of participants that they take these tests from. And this is from a MESA study, so this is I think they did this over 10 years, and they ended up measuring the intima thickness over 10 years. Okay, so here we've got our large LDL-P, so the nanomole per liter. We've got low levels, so in the white bars, we've got low levels of large LDL. These grayish bars, we've got a little bit higher, and then the black is really high levels of LDL-P. On this axis right here, we have all of these are grouped together, are with small LDL-P. So there's low levels of small LDL-P. So just to orient you, so this white bar in this condition right here is low levels of small LDL-P and low levels of large LDL-P. So this is the amount of intima thickness that there is there's only 10 individuals that have both of those at the same time. So it's a pretty small N. And let's say it's around 900. And all of these are relatively low. So if you have large LDL-P, you have maybe a slightly elevated level, even though you have slightly more, a thicker intima thickness, even though you have small LDL-P. And you can see that continuously. So as small LDL-P increases, the combination of large LDL-P plus the small LDL-P leads to substantial increases in intima thickness. So that combination, having both of them elevated, not good. But then you got to think, if that's the case, most likely, depending on the ratio, most likely you have really high LDL particles as a whole, not just because, again, the small and the large combine to make the total LDL-P. So we see that there's really high risk. And again, the sample size here is really small. But overall, you can still see this trend that they start to increase as small LDL-P levels increase. Now, additionally to that, in all the conditions that you have large LDL-P, you still have greater intima thickness. So even if you were to look at, like by comparison to having like lower levels of LDL-P or large LDL-P. So no matter what, there's probably still an increased risk, but we're going to get deeper into that. But the big point here is that it definitely tracks with small LDL-P for sure. So then they quantified the relationship. There's more data to this study than I'm showing, but I'm showing you the highlights that indicate they look at some other factors as well, and you can certainly look at it for yourself. I'll have it listed for you. But I'm pointing out specifically the interactions that they're looking at for small versus large. So again, looking at intima thickness, looking at the large LDL-P in quintile, so this is the lowest level, so in white, and then the black is the largest amount. And they adjust it for age and for sex, but they don't control for small LDL-P. They see that there's no relationship, so that there's no increase in intima thickness when you just look at the large LDL-P. However, when you do adjust for the small LDL particles, suddenly you see that there is an increased risk, and then it really shoots up when you have really high levels of LDL-P. Now, one of the problems with this still is that this could still be just feeding into having higher total LDL particles. So what they did is they did a further analysis. And we're going to focus on these two up here, large LDL particles, although they also look at LDL cholesterol. Remember, there's a distinction there. This is not LDL-P, this is LDL-C and HDL-C and triglycerides. But they've got three different models. They've got Model 1, Model 2, and Model 3. The large LDL-P, so essentially any number that's any number here is essentially increased risk if there's a statistical significance. So anything above 0 would be, you're getting closer and closer into a statistically significant increased risk of thicker intima thickness, which is a proxy for cardiovascular disease risk. Now, you could debate that, but I think that's pretty sound evidence. Although it'd be nice for them to also do this kind of analysis in specifically heart attacks or cardiovascular disease, like some specific endpoints, or to look at death, cardiovascular disease related death, or all-cause mortality. That would also be interesting. Although now you're getting a lot more other variables involved. Anyway, I digress. The point is, they're looking at Model 1. So this is adjusted for age, sex, smoking, race, and hypertension, which is nice. And they find that there is probably an increased risk with the small LDL particles compared to the large LDL. Now, keep in mind, they're not actually doing statistics between these two. So this is just my eye. If you disagree, please, that's perfectly all right. But both of them have increased risk. But the small LDL-P have even more increased risk. Then Model 2, where they're actually also looking, so they're looking at all the same factors plus looking at LDL-C. So looking at the total LDL cholesterol levels. And you can see that suddenly the values decrease, especially in the small LDL particles. It decreases, but it's still very statistically significant. And you still see that there's a slight increase in the small LDL-P versus the large LDL-P. Now, when we look at all of this adjusted plus HDL-C plus triglycerides, we see that there's still an elevated risk, but there's probably no difference between these two. So we went from thinking that small LDL particles are more detrimental than large LDL particles to now the analysis, once you start adjusting for all these other factors, confounding factors, there doesn't seem to be much of a difference between the two. Now, they're also adjusting only for LDL-C and not LDL-P, which would have been really interesting to see if they can maybe nullify this entire effect. And then we would be able to tell, is the entire effect driven only by LDL particles and not by the size of the particles? So this is just one study, and I would like to, there's certain drawbacks of this study. So like it's an associative study, totally get that. Even with some pretty powerful statistical means to try to to remove certain factors out of the equation to be able to tell what's the association of just this factor versus this factor. Additionally, they're looking at IMT. They're only looking at intima thickness. So that's a drawback. And it doesn't completely convince me that there's no difference between small LDL particles and large LDL particles, or specifically that because we need more data in general, more than just one study, but it at least swings it that way. And small LDL particles, we don't know if it has an independent risk independent of LDL particles in total. So those are questions that still need to be answered. Now, that said, there's also studies, so this same study and some of the other studies as well mentioned that people that have hypercholesterolemia, which means that they have genetically, so as opposed to the other people that had the mutations that reduce their cholesterol levels, there are some people that have elevated cholesterol levels from essentially birth. So they just have continuously high LDL cholesterol. And those cholesterol particles tend to only be of the large variant. And these studies, almost all of them indicate that there's increased cardiovascular disease risk. So my point being, the idea that large buoyant LDL particles are not a cardiovascular risk factor is nonsense, because it's still a factor. It still has an effect, because there are also some mechanisms for why it is detrimental that small dense LDL particles don't do, which I won't go into for the time being. But, well, one of them is like cholesterol crystals. It contains more cholesterol. So when they get damaged, they get taken up into the intima. They get damaged, they break open, and then you have cholesterol crystals that get taken up by macrophages, so immune cells, and then ultimately that leads to foam cells, and you also get a greater inflammatory response from the large LDL based on that mechanism. So not looking at the oxidation mechanism. Anyway, I'm nerding out a little bit. But the point is that you have multiple studies. So you have this association, obviously, but you also have genetic studies that look at people that have highly expressed throughout their entire lives. They lead shorter lives, typically. They have more cardiovascular disease events. And they, I wouldn't say exclusively, but they dominantly produce large buoyant LDL. So really, if you just have high LDL, you're at risk. That's what I'm getting at. So the conclusion up to this point, and next what we're going to do for the insiders is, for the full version of this video, we're going to go into different techniques for lowering LDL as well as small dense LDL, because we still don't really know if small dense LDL have an independent risk that's independent of the total LDL. Also looking at a threshold. So one of the reviews specifically mentioned a threshold of risk. So if you reach a certain small dense LDL threshold, that if you're under that, you may actually be okay, that there isn't a negative effect. And then some genes. So if you wanted to do like a 23andMe or I don't know, whatever those genetic tests are, you can actually look at the raw data and you can look for particular genes. So I'm going to give you some of those genes, and because these genes are implicated and associated with elevated levels of specifically small dense LDL. So if you want to find out about that. And then a triglyceride effect, which I find really, really fascinating. I'll go into some of the physiology on that. Okay, but that's for the full version. Up to now, though, what's kind of the takeaways up to now? For sure, the higher your blood LDL particle levels, the greater your risk of heart disease. There may be a slightly added risk if LDL particles are small, dense in nature, but this has yet to be confirmed. This is not set in stone, but that is yet to be defined as an independent factor like total LDL particles. So if you control for LDL particles and you have high small dense LDL particles, do you still have increased risk? However, even large buoyant LDL particles do increase heart disease risk. So regardless of the type, the more LDL particles, the higher your risk. Additionally, this is a big takeaway. Additionally, opting for particle tests, so LDL particles as opposed to LDL-C, which is what we typically get, over cholesterol tests is I would say far more accurate measure of your health state. So I hope that you learned something from this. This was so fascinating for me. I had no idea where the data was going to go with this. I had heard obviously about LDL and all that stuff, but I hadn't heard about the different sizes of LDL. So I'm educating myself on all that stuff. It was really intriguing, really fascinating. And I hope that you learned quite a lot about it. I left out so much more information on the mechanisms of how inflammation gets involved and stuff. And I will be going into that stuff in future content, but I just wanted to specifically address small dense LDL, large buoyant LDL, and total LDL, and LDL-C versus LDL-P. I hope this was informative, and with that, thanks for listening up to this point, and I'll hope to have the pleasure of speaking with you in the future. Bye.